No, but that's the one at the top. But then what's the one underneath? Are they
It
that?
rotates.
I don't know if the eyes.
No, it's all the things for the image based on.
The eyes, maybe.
He's a clever little boy. Must be protected at all costs.
We don't have to use one before, can you?
Can you hear us and see us, Chris?
Yeah, that's fine.
Good.
So how were the tutorials? How far did you get with SnakeMake?
We just did an instruction. We spent I spent most of the time this morning on explaining the rest of yesterday's tutorial. So we just spent 30 minutes on SnakeMake, but I think it's already good because now we got another view of SnakeMake.
Okay, so you didn't get them to construct their own pipeline for binning, right?
No.
Which is the next step of that tutorial.
Yes.
Okay.
Not yet.
So I think it's probably worth doing that, isn't it? So probably that's what you should do this afternoon. Do you think?
I don't know. Do you guys want to build your own pipeline, or would you prefer starting something different, not strain resolution? Do you have any strong opinion on that?
Well,
I mean, like,
If the pipeline will be useful, probably good to do.
but then is that going to mean we're not going to get any of the other stuff that we're meant to get done today? Because that
no,
will
what
take
we
us all
might
afternoon.
end up doing is then doing some of that on Thursday.
So shift things.
So I think we'd still do the strain resolution, but maybe do that on Thursday and cut out some of Thursday and Friday's material. So there might be less statistics on Friday, or maybe less machine learning on Thursday.
Chris, it's already it's too late. You already sold less statistics to people. Now they are happy.
But I'm still writing those tutorials, so I'm sort of keen to reduce their size. That's fine.
Yeah.
Well, I mean, I think both of these things are useful, but I sort of feel that building your own SnakeMake pipeline is one of the most useful things, isn't it?
Yeah.
Because if they're ever going to try and do something of their own, rather than just running our pipelines.
Yeah.
So I think we should do that first.
How does it decide who to look at?
It's
The noise.
Oh, it's because you laughed.
Yeah.
All right. Well, look, I definitely. Sorry, what's up?
Pardon?
We're fascinated by the owl.
I'm confused.
Well, the owl, it moves its eyes around and watches. Look, it's looking at me now. If Harshni, say something at the back. Come on.
He's
All right.
It's too far away to focus on
a
it.
bit loud.
There
Yeah.
you
All right. Well, anyway, so I'll do the strain resolution lecture now, but I certainly think you should do some of the SnakeMake tutorial. So,
Okay.
So yeah. All right, but we can also see what people think after I've gone through. Yeah. Okay. All right. So I'll start sharing my screen then. And in a way, this is like even more complex. If you thought the binning was complicated, this is where it starts to get properly complicated, right? So yeah, I do wonder if it's better to focus on getting some of the more straightforward things well understood. All right. Anyway, I'm going to talk about some of the pipelines we've developed for actually going to finer scale resolution than simply mags, and I'll explain what I mean by that. So the basic idea here is to try and get down to the level of strains in microbial communities, but there is a philosophical question here around what do we actually mean by a strain, and actually Nick Lohman once basically said, my definition of a strain is whatever my program results. Okay, which is sort of how we feel about things, right? You know, if the program gives you the answer, that's a strain. But anyway, so I'll try and explain what, you know, the programs we're talking about today, they're trying to generate. Okay, so this is quite an old review paper on strains and species and genomes from 2005, but it's still quite useful, and it sort of introduced a lot of the, I suppose, the ideas that we have today. So what they've done here on the x-axis is average nucleotide identity. Okay, so you find that by comparing. In fact, you know how to do that, right? Because you did that in comparative genomics. So how would I compute average nucleotide identity between two genomes? Anybody?
Did you do that last week? ANI calculations?
No.
Probably not. I don't think they did. They
right.
called out SNPs and
Okay.
stuff, but they didn't actually do ANI.
Oh, right, really? I mean, for me, that's a large part of comparative genomics. Anyway, it's a very useful thing to do with genome collections or MAG collections. Okay. The basic principle here is you compute average nucleotide identity on the shared portions of the genomes. So if we take two strains of E. coli, okay, some of their genomes will be shared, and some of them will be different in terms at the gene level. Okay, on those shared portions of the genomes, which will include those core genes that we've been talking about. Okay, but we'll have other genes as well because there's many genes that are core to E. coli. All right. We can calculate average nucleotide identity. So the numbers of SNPs per, basically the SNP frequency. Okay. But we can also calculate how much of the genome differs. And here they've done it in terms of megabase pairs. So this is between those two genomes, what's the total length of the bit that doesn't overlap? Okay? And the two things correlate. This is across a wide range of species. The further they are apart in average nucleotide identity, the more the less of the genome is shared. In practice, you would do this. There is a very fast, there's a program called Fast ANI for doing the nucleotide identity. Or if you wanted to do it more carefully, you can use programs like MOVE to align two genomes together. Okay, so the two things correlate, and there's a sort of gap around 95%, although I do notice here that it does actually overlie two of the data points. So you've got to wonder how good that gap actually is. And that's what is typically taken as the definition of species based on genome identity. So if two things are less than 5% divergence or more than 95% similar, we assign them to the same species. So all these E. coli and Shigella, and this is one of the reasons why Shigella is really the same species as E. coli, they all lie within that 95%. Okay, but we've got variation below that 95%, and that's basically what we're going to try and deal with today. Can we resolve some of that variation directly out of metagenomes in a de novo way? Okay. Now, what we define by a strain, though, is not what a typical microbial, say a typical clinical microbiologist or an experimental microbiologist might consider a strain. So they basically consider every single isolate a different strain. That's sort of reasonable up to a point. I mean, if you've got an epidemic, things will be almost identical, but I suppose there might be like one SNP difference on every genome you isolate, possibly. Instead, we're going to try and aim to resolve, so that's not our definition of our strain. We're basically trying to resolve subpopulations within species that are ecologically coherent. Okay, so basically within this 95% definition, we may have subpopulations with different population dynamics and different ecological roles, and different genomes, and can we resolve those genomes? Yeah, and you can think about what would sort of generate such observations. It would be things like clonal expansions, but also ecology, niche selection, but then things like recombination and migration would dilute these coherent subpopulations. And these concepts are nicely explained in this O'Brien paper in 2014, where they sort of define these things as lineages, actually, which is a term that's starting to become popular again with now we can get high resolution genomes out of long read data. People are starting to call them lineages. So it's sort of like a group of related organisms that form a taxa that is, as I said, ecologically distinct from other members of the same species. Okay, so hopefully that helps explain what we at least mean by a strain in this context. Now you could do these types of analysis either in a de novo strategy or reference based, and we talked about that a bit yesterday in the context of 16S and metagenomics. I'm going to explain what the difference between those two are in the concept of strain resolution, but then I'll talk about our two pipelines for de novo strain resolution. Actually, that will be on Thursday, not today. So yeah, sorry, that wasn't corrected from the last version of this talk. All right. So what's the difference between de novo and reference-based strain resolution? Well, it's just the same as when you're doing OTUs or doing classification of 16S reads. When you're doing classification of 16S reads, you're comparing to a big database. When we're doing reference-based strain resolution, we'd compare to a big database of genomes. So the Enterobase in Warwick, where me and Mark used to work, this has got some of the largest databases for various pathogenic organisms. They've now got over 200,000 salmonella genomes in that database. For those types of organisms, a reference-based approach makes sense. But then there's a lot of organisms where we don't have such large-scale strain databases. And as I sort of mentioned yesterday, I sort of fundamentally feel that, at least for the human gut microbiome, that in every individual there is a unique strain of at least some organisms. So effectively, we're always going to need de novo methods. Yeah. So how, given a database, could you find the strain profiles? Well, yeah. Again, it's a bit similar to what I was saying yesterday. There's two strategies. One that's adopted by Metaflan is basically the same as Metaflan for species, but they go on and try to define genes that are unique to strains and profile metagenomes in that way. I think that's probably a bad idea because a lot of those anything that is unique to a strain on a gene level is likely to be a gene that is definitely accessory and also likely to move quite easily between strains. So I'm not quite convinced by that strategy, and actually it's not often used to a fine scale. It's probably better to use SNVs on those shared genes that I was talking about, and that's the approach adopted by this Albanese paper. They're basically, so each strain has a different profile of SNPs on the core genes. What you observe in your metagenome will be a mixture of those SNP frequencies, depending on the proportion of the different strains that are actually present, and then you try to deconvolute that mixture. And probably things, and actually a similar strategy is adopted by MSweep, which came out pretty recently. And yeah, I probably think that those SNP profiles on core genes is probably a better strategy overall for reference-based. All right, so that's reference-based strain resolution. Was that all clear? Do you have any questions on that? before I move on to sort of the de novo stuff? All right, okay. Right, so what we do is de novo strain resolution. As I said, it's more difficult, but it has the advantage of wider sort of applicability. And our starting point then is not actually reference genomes, but MAGs, okay? So the principle here is that if you have constructed a MAG from short reads, and possibly actually we're finding out some of the time from long reads, then it's going to be a composite of many genomes. And this is something that is important for you to bear in mind, even if you're not interested in strain resolution. Even if your only interest is MAGs, it's important to realise that there's more than one genome represented in that MAG. And that variation will be of two types, just as we've been talking about. It will be SNPs on core shared genes, and it will also correspond to variations in the accessory genome. So, we can see in terms of SNPs, you can find this quite easily just by mapping reads back from different samples. So say we've done a co-assembly and we map all the reads back from one sample, we might see an A at one position because the strain that's dominant in that sample has that sequence. Whereas if we map back a different sample, we'll see different SNPs at those variable positions. So that's the two sources of strain variation. Now, what we use in our first pipeline, Desmond, is basically those SNP frequencies on the shared genes. So the principle to the algorithm is just to map, as I've just explained, we map the reads back from samples separately. We look for variant positions, and we determine the frequencies of the bases at those variant positions. As there are different ways to do this, based on just a single sample. That basically comes down to what is called read phasing. So that's where you look for shared SNPs that are shared in the same read. So Strain Express is the most recent algorithm to do this. That will work for very divergent strains, but it's not going to work for things that are quite similar, because you won't be able to chain the SNPs together across large parts of the genome, basically, because the reads are short. I mean, if you could, you would have actually assembled out the two strains separately to begin with. So as I said, we use these SNP frequencies on these variant positions to try to resolve the strains. We implemented that in this pipeline we call Desmond for de novo extraction of strains from metagenomes. And I'll just briefly explain the principles behind that to you without going into the sort of technical details. But basically, it's a lot like the Contig clustering I talked about yesterday. So here I'm showing just for a single gene the proportions of the minor SNP at all the variant positions for this gene. And this was a synthetic example where we had 5 different strains present across 64 different samples. So that's the columns of this heat map. The rows are then the SNP positions. Okay. So you can see that you see similar SNP abundance profiles across the samples. Okay, and that's because all these SNPs here are found in this particular strain, haplotype 2. Okay. And then similarly, all these SNPs are found in haplotype 3. Okay, so we can link those together. And this was actually first done in a program called Constrains, but you have to be careful how you do it. Okay, you can't just simply link the SNPs together because of things like this. So here, position 217, we see a profile that's clearly the sum of H2 and H3. And that's because both of those haplotypes share that SNP. So it's not a simple clustering, it's actually it's more of like a deconvolution. It's quite similar to algorithms like non-negative matrix factorization, which try to decompose matrix into sums. So the way to think of this is that each strain forms a signature with a unique set of abundances, and we're trying to deconvolve those signatures. It's actually just the same as the Albanese algorithm for reference base, except for they know the answer, okay? So if you know the SNPs, it's quite easy to find the abundance profiles across samples, but we don't know the SNPs, so we're trying to do the two things simultaneously, basically. Okay, I'll sort of return to that when we talk about our more recent algorithm, Strong, so hopefully that will become clear. But this is an overview of the entire Desmond pipeline. So the first parts are what you're familiar with now. We get separate samples, do a co-assembly, generate bins, and then identify single copy core genes, and then we find the variant positions on those single copy core genes, and using that we're able to deconvolve the haplotypes for the different strains, and then also their abundances across samples. There's always the question of how many haplotypes are present. So here we use quite a simple strategy where we look at the quality of the fit in terms of this quantity called deviance, and we look at how it improves as you increase the number of strains, and it always will improve. That's sort of the nature of these types of algorithms. And then when it stops improving significantly, we decide we've got enough strains. So it's sort of a little bit arbitrary, although I did introduce a sort of automatic heuristic for doing that. We have better ways to do that now. And then finally, once we've got these haplotype profiles, you can actually get the accessory genes in the different strains by looking at the coverage profiles of those accessory genes and trying to explain them using the coverage profiles of the different strains. Okay, so that's the Desmond algorithm. I'll just briefly go through the application of it that we, one of the applications that we applied it to in the paper. And this was a data set published in 2015 from, this was the first global survey of the oceans. That was TARA, Ocean Planktonic Metagenomes. That was TARA. And actually collaboration, well, it was Tom Delmont, somebody we were working with at the time. He did a mag collection from TARA, which is actually one of the, well, it was not one of, it was the first global ocean mag collection. So he got 660 different mags from TARA Oceans. And this is a tree combining those with reference genomes. So we had an interesting cross-section of planktonic diversity. Here we've got Archaea. Over there, this would be SAR11 type groups. So these are these highly streamlined organisms. So the ocean is amazing because you get some of the smallest free-living bacterial genomes known. So Pelagibacter ubiqua is about 900 kilobase pairs, I think. And this is also probably, I think it's probably the single most numerically abundant bacteria. I believe that would be probably true. It's everywhere in vast numbers, hence the name. It's also known as SAR11, right? So anyway, so we've got a nice set of mags. So we applied Desmond to these mags. You can only really run it when you've got sufficient coverage, because you need those SNP frequencies. So we only applied it to 32 mags from TARA, where we had a total coverage across all samples greater than 100. And one simple thing that we observed is when we looked at the SNP frequency on those core genes was a nice negative correlation with the metabolic complexity of the genome. So it seems most of the SNP diversity is in the simpler, streamlined genomes. So Desmond then allows us to deconvolve that diversity, and I'm just showing you an example from one MAG here. So this MAG we successfully resolved into 3 different haplotypes. So the MAG was one of these streamlined gamma proteobacteria, and this is showing its abundance across the whole ocean. And what's nice is that the different haplotypes have different, are regionally specialised. So haplotype 2 here was found with higher abundance in the Mediterranean, haplotype 4 in the Indian Ocean, haplotype 3 in the Pacific. And actually that was true in general across all the Tara mags that we resolved into strains. Okay. Then the other thing we did actually was to look at the accessory genome. So this is what I'm showing on this plot here. Just to say, when we look at the number of haplotypes, there's a negative correlation with genome length. So most of the strain diversity, which sort of explains that SNP diversity we're seeing, is in those small, those streamlined organisms with genomes around 1 megabase pair. And as you get to bigger genomes, we were observing less strains in general. That's not good, is it? Right. Now, this plot here, though, is basically doing the same analysis as in that paper I showed you in the introduction. We've got, for each now pair of strains from the same mag, I've computed their nucleotide divergence in terms of SNPs, so that these are 2.5% difference on shared genes or SNPs, which I'm showing on the x-axis. And then on the y-axis, I used the second part of Desmond to find the accessory genomes, which means I can compute how divergent they are in terms of accessory genomes. And the two things correlate, which is sort of reassuring because it's what you'd expect. The more different the pair of strains are, the more different their accessory genomes. But what was quite interesting, and something one day I hope to sort of return to, is that the strength of that correlation seemed to be stronger for the very small genomes. So the slope of that line was significantly greater than that line. So the blue dots here are all for all mags with a genome less than 1 megabase pair. So it's possible that this is due to niche differentiation. So if you have a large genome, you may be able to move into a new niche with less actual change in genome content because you can have more change in expression levels. Whereas if you're one of these streamlined genomes where essentially sort of every gene has to be used, then for niche differentiation, you need a larger amount of, a larger change in the accessory genome. So anyway, so that's, hopefully giving an example of the sort of thing you can do with Desmond, and it has been applied quite a few studies now. I think the one I found most interesting was somebody used it to resolve strains in the symbionts of mussels that live in deep sea vents. All right, so I'm now going to talk about our sort of update to that algorithm, which we call STRONG. And this is basically why I talked so much about assembly graphs yesterday, so that hopefully this would make sense to you. So if you remember, an assembly graph actually encapsulates the possible variation in a sequence. But what you do when you construct contigs is you simplify all that variation. Okay, so actually all the information we need to find strains is already in the assembly graph. So in practice, you don't need to take the contigs and map the reads back. Instead, you could work directly with the graph itself prior to that simplification. And that has a lot of potential advantages. You don't need to map reads anymore. Instead, because all the variation is in the graph, you can use something called threading, which is far simpler. You just do exact k-mer matching, actually. And it also allows a more complex variant structure, so you can have things like whole gene insertions. And it incorporates some of that read phasing information as well, because in some instances, if your assembly graph, you'll be able to incorporate phasing up to the length of the k-mer used to build a De Bruijn graph, for instance. So it has a number of advantages, and this is actually pointed out in a paper by Titus Brown. But they pointed out that assembly graphs would be really helpful for resolving strains. They didn't really actually have an algorithm for doing it. So this is what we do in the strong algorithm, and it's essentially the same principles as Desmond. But the input now is a graph, and with the coverage of each node of that graph across multiple samples. So this corresponds to, say, node 13, Unitig 13, and this is its coverage profile across 10 different samples. So we're trying to, this is what we're trying to explain, and what we're trying to find is the paths through that graph and the abundances of the haplotypes that best fit the input data, which is those coverages and the graphs. Now, I think I just put this in to horrify you. This is the actual mathematics behind it. If you're interested in that, then I can go through this on Thursday when I'm here in person. But yeah, I think but essentially we have a Gaussian distribution on each node, and we're trying to explain that in the same principle as Desmond, where the expected coverage is the sum over the strain coverages that pass through the corresponding node. That's the principles behind it. And then we use the same type of algorithm that we use in Concoct, in fact. It's a variational inference algorithm that iterates to the best solution that explains, in terms of finding the paths, that explains those coverages. The overall pipeline, though, is very much like Desmond. We build mags, but the key thing here is that we pull out that high resolution graph with all the variation in, and then we extract subgraphs from that corresponding to the single copy core genes that we can then resolve our strains on. So it's a complex pipeline, and we'll do our best to show you some of it this afternoon. But a lot of the components of the pipeline, pulling out the subgraphs and getting their coverages, are sort of super useful in other contexts. So actually, I just saw that somebody published a pipeline for doing something similar with prophages, and they've used some of the components of Strong to do that. So hopefully we will have time to show you some of these things, because it's just generally useful to be able to work with assembly graphs. So one issue we do have in Strong is noise when you have more higher coverage organisms. And so we've got this very simple simplification procedure based on estimating local coverage and removing any node that has a coverage below some local threshold that really nicely simplifies the graphs. This was all done by somebody called Sergei Nerk, who is one of the Spades developers. Here is how it does on some synthetic data. So the synthetic data was a 45 species community. 20 species were present in one strain. I think 10 with 2, 5 with 3, 5 with 4, and 5 species with 5 strains. And this is the results from Desmond on the top row. Mixture S was the only competing algorithm at the time, and here is Strong at the bottom. And each of these figures is showing actual strain number on the x-axis versus predicted strain number on the y-axis. So a lot of the time, well, a good proportion of the time Strong gets it right, at least when you have a reasonable number of samples. So this is a result for 10 samples. It does somewhat less well with just 3 samples, but even when it doesn't get exactly the right strain number, it at least gets most of the strains that are present. Desmond, yeah, not so good, and that's probably, it has a less sophisticated way of determining strain number, plus all those advantages about using the graph. And Mixture S, which is a sort of phasing EM type algorithm, is actually quite awful, so I won't even talk about that. and then the strains that it does predict, at least when you have reasonable coverage, so above 20, are predicted actually surprisingly accurately. And we've started to get results from long reads now, so we can sort of directly compare strong to long read haplotypes. And yeah, it's actually very encouraging on the whole. All right, so I'm just going to conclude with some examples from actual applications of strong. How am I doing for time? Because I can either go through both or just one of these.
You've been 38 minutes so far, or so.
All right, okay. I'll see how it goes. I might just talk about the FMT, because most people here are interested in the human microbiome, aren't they? Is there anybody doing non-human microbiome stuff?
No.
All right, I'll just talk about the FMT to give you time to get onto your tutorials. Okay. It's quite a nice example anyway. So this is a fecal microbiome transplant data set generated by by my collaborator Meron, who is now in Germany actually, and his student Andrea Watson. So this is a nice data set because they have a time series of the donor for a year prior to the FMT, and they have a time series of the recipient for a year post FMT, and then they also have 2 time points pre-FMT for the recipient. Now, I'm just looking at one individual here from the study. This person, they were receiving treatment for C. difficile, and here I've constructed MAGs, and the heat maps are showing you the MAG abundances across both the donor and the recipient. So what's noticeable is the recipient has very few MAGs. Okay, so it's a very dysbiotic microbiome. We've actually only got three or so MAGs that are turning up, maybe a couple there as well. The donor has a nice complex microbiome, as you would hope, and then you can sort of see that post-FMT, the recipient microbiome looks complex as well. So in that sense, it looks like the FMT has worked, and the person did actually get better. But what
So
I
that
did
red
here
thing,
was
is
to
that
look at
C.
the
diff?
strain dynamics.
Is that red thing
Sorry?
C. diff? The red thing, is that C. diff?
No, I think that's actually the one I'm going to talk about. This one here, C. diff. Yeah, it's a good point actually. I don't think C. diff was picked up with this level of sequencing. I think that's true. I should really check that. It's a good point. So this is the problem with never really thinking about clinical microbiology. I just concern myself with the commensal organisms, right? But yeah, it's a good point. Okay, so we did a co-assembly of all these samples together. It's actually a pretty good co-assembly. We'll talk more about assembly on Thursday. So the M50 was 10 kilobase pairs. That's the sort of typical length of a contig. And we've got 109 mags in total. So that's what I'm showing the abundance of here. Actually, possibly it could be C. diff. So I was a bit confused there. I thought I will have a look at that actually, Mark, and see if it is in the data set. But I'm going to focus on this mag here, in fact. The reason for that is because it's quite an interesting one, because it is present in the recipient prior to the FMT, and it's still present post-FMT. So it's quite an interesting organism to look at the strain dynamics in. And in this case, we're able to resolve 2 strains with strong. One is associated with the donor. So this is showing the donor strain proportions in that year preceding the FMT. And one was associated with the recipient FMT. Post-FMT, that is replaced by the donor strain. But what's quite interesting is that over the following year, the abundance, well, basically the recipient strain that looked like it had been eliminated bounces back and comes to actually form the majority of the population of that species. So yeah, it might appear superficially in terms of MAG that the FMT has worked, but at least in terms of strains, it perhaps it only works temporarily. Although it is actually sort of more interesting than that, because one thing we also managed to show is that this recipient strain isn't quite the same as it was. All right, so there is a prophage that moves into the recipient strain that was carried on the donor strain. So anyway, it's quite an interesting example of strain dynamics, and I went on and had a look at the rest of the mags in this analysis. So here is an example where we actually had quite a lot of diversity. This was a Blautia species. So here Strong was able to resolve 4 different strains, and this is just showing the haplotype paths through one of those cogs. So the dynamics in the donor now were somewhat more complicated. You had this strain in green was fairly low abundance, and then these strains were fluctuating a bit over time. But what's quite clear is that, and this was a case where there was none of this mag in the recipient pre-FMT. So basically, post-FMT, it's got a profile that looks a lot like the donors at the point of the FMT, but then over time, actually, the other 3 of the strains basically seem to get eliminated. Maybe this red one is still there after the year, but it becomes dominated by one of the donor strains. There's clearly a lot more going on at the strain level than perhaps just at the mag level. And to sort of summarize that, I looked at all the mags. Okay, so each point here is a single mag, and I've computed the correlation in strain proportions between the donor and the recipient immediately post FMT. Okay, so there's a good correlation between the two, suggesting that for most mags, the strains transplant along with the mags. Okay, but then a year later, that correlation is much weaker. So over time, some of the donor strains are being lost, the strain proportions are changing. Obviously, you would expect to see that anyway. So one way we can sort of correlate that, quantify that is, so the red line here is over the time showing the breakdown in average correlation across all mags for the patient, recipient, and then the blue line is the same for the donor, but going back in time, basically. So we get a sort of baseline. So yeah, going back that way in terms of the time points. So in almost entirely, you can see that the correlation within the donor at equivalent timescale is greater than it is for the recipient. So it sort of shows the effect of, there is some localized ecology going on in the recipient crop, which means the strains aren't being transplanted, except for this time point here, where the entire sort of strain spectrum of the donor gets upset. So this could correlate with them being ill. There might be some sort of infection or something. But it also sort of illustrates that I didn't make the results up. If you've got one result, one time point that doesn't work, and you can do this, so you can do this with large-scale culturing, but these bioinformatic methods are perhaps a more convenient, higher throughput way to understand strain dynamics in FMT. All right, so look, I'll conclude there because my other example was based on those AD examples, and I think that's perhaps less relevant to you. So I hope I've showed how we can use time series to deconvolve strains de novo. And as I said, Desmond is our first iteration of this. It's probably still useful in some contexts, even though, as I said, Strong seems to work better. The context where it's perhaps more useful than Strong are cross-sectional studies. Okay, so Strong really does assume that you've got these definitely distinct subpopulations. I think Desmond will still work even when you're not so much finding true strains as ecological patterns, and that's possibly what's going the Tara data set. And it helps us understand this within species niche partitioning. All right. right. So look, I'll stop there so that you can get on with the tutorials again. And were there any questions? I know that's quite a sort of fairly complex set of ideas.
But
yeah,
you've been quite provocative at the end by saying you explain niche partitioning, but you haven't. What is the basis of those changes between the strains? Is there any functional difference between the strains that we can say, that one has got that particular metabolic pathway that allows it to live into this circumstance, or that one has got a type 6 secretion system which that one hasn't got?
Yeah, okay. So partly that came down to what I didn't have time to talk about.
Right.
So what we see in the anaerobic digesters, and this is admittedly it's a hypothesis, okay? We see far more stability in strain proportions than we see in mag proportions, okay? So remember the AD example, we had large shifts in the community structure, okay, at the mag level. If you look within the mags, actually there's less shift in the strain proportions. So to me, that is evidence of within the species niche partitioning for the strains. If they were all doing the same thing, then I don't see why their proportions would stay constant.
Okay.
And we saw a similar thing in the oceans as well, actually.
But you should be able to.
So yeah, that was.
But you can identify the accessory genome differences, the differences in gene sets, and say, look, they've got 100 genes in that strain that not in that strain. Can we predict any phenotypic consequence of that? Have you tried that, or that's too hard a question?
No, it's not too hard a question. What we could do with Strong is take the accessory genome prediction algorithm from Desmond and use it with Strong haplotypes. One of the reasons we haven't done that is we now actually have a load of PacBio data from the AD, which will give us perfect accessory genomes. So, but we should do it and compare to the PacBio genomes. I think that's the right thing to do. But no, it's a good point, and it's something that, I think is worth more exploration. And then also to actually look at transcriptomics data as well would be awesome in this context. Because you could not just see whether the genes are different, but whether they're actually being expressed differently as well between the two strains. So no, these are all interesting questions.
Any other questions?
Chris, I have a question for you.
Yeah.
Can you tell me again what are the sort of outputs of Desmond or Strong? Because you talk about strain, but what do we get in the end?
Okay, that is a good question, Seb. Although I think you probably know the answer.
Oh no, it's been so long. I don't know at all.
So yeah, you get two things basically. You get the strain proportions across samples, which is what you use to get those dynamics. That's what I was plotting in those graphs there. And then you also get the haplotypes on the core genes that you can use to build phylogenies and determine how similar your strains are in terms of nucleotide sequence. And then with Desmond, then you also get the accessory genomes. But as I said, we haven't actually implemented that bit in Strong yet. It's probably something we should do next year. So anyway, we talked about it, didn't we? All right. Okay. Well, look, I think that I'll let you get on now with the tutorials.
Okay.
And I'm hoping. Unless I don't, I'm sort of feeling a bit better, and I'm hoping I'll be in person on Thursday. Although Seb, do you think you'll get onto the Strong tutorial today? I suppose you can just do it yourself, though, can't you? Right?
Pardon?
Maybe that's easier.
You can
Are
do
you
the
okay
Strong
to do
tutorial.
the Strong tutorial if you have time to get to it today?
Yes, sure.
Okay. All right. Okay. Well, thanks a lot. It's nice talking to you all again. Bye bye.
See you then.
Bye.